Wait for it, little spinny ball. There we go. All right, we're back. We'll see if we can finish off the platyhelminthes today. So I'll go ahead and start the slides up. And we'll move to the last groups of platyhelminthes I want to work on. Monogeneans. And cestodes. That is, monogeneans are this parasitic group that doesn't infect humans that you may not know about. And cestodes are the tapeworms, which certainly do infect humans. Most commonly live on fish. There are some that will parasitize. When I was looking for ideas for labs for this course, sorry, I don't know why that happened. Hang on. That's not supposed to be happening. Anyway, when I was looking for labs for this course, I came across an old, 1940s era zoology textbook, which are sometimes worth looking at because they can have interesting ideas. And the problem is that one of them, am I still presenting? Hang on. So they sometimes have really nifty ideas for lab that have, sometimes been overlooked. But one of them had suggestions for labs you could do that involved monogeneans. And the suggestion was that you should just go out to a pond. There, sorry. I killed off the rim shots. You should just go out to a pond and collect a whole bunch of turtles and kill them and open their mouths up. And there was a particularly common monogenean species that you could often find, which may have been the sort of thing you could get away with in the 40s, but we can't really do that now for reasons that I hope are obvious. So yeah, we're not going to go kill 30 turtles to go look for monogeneans in their mouths, but they do sometimes infect turtles, frog bladders, and there's one that has colonized the eye sockets of hippopotamus because somebody has to. Life finds a way to occupy some of the weirdest niches you can think of. And yes, there is a worm that parasitizes hippo eye sockets. They feed on blood, mucus, or tissues. Most don't do a lot of harm. There are a couple of species that can cause damage to farmed fish stocks, so they can infect or affect the human economy. The life cycle is pretty simple. You don't get asexual reproduction. The eggs just hatch into a ciliated larva that swims away and finds new hosts. And here's what these look like. They're all parasites either on the outside of their host or in organs that open to the outside. So you'll get them on fish gills. You'll get them inside bladders. You might get them up rectums or places like that. And you might get them in eye sockets, as I mentioned. But you're not going to find them burrowing deep into the liver or living in the bloodstream or something like that. They're always found in body parts that are open to the outside. Now, in this one, the mouth is over to the right. The mouth is surrounded by a set of suckers and pads that make up what's called the prohaptor. Prohaptor means front grabber. Inside the mouth, there is a sucking pharynx. And over on the left, you've got that big round disc-like thing that looks like a suction cup. That's just called the haptor. Sometimes you'll see it called the opisthaptor, the grabber behind, as opposed to the prohaptor, the grabber in front. And in this particular species, there's a pair of anchors in the haptor. They're just hooks that hook onto the tissues of the host. The fancy name for these hooks is the hamuli. There's another one, and in this one, the mouth is on the left. The prohaptor is right there. The mouth leads into this highly branched gut. That's most of the branchy, mossy-looking stuff that you see. And in this species, the opisthaptor is off to the right, and instead of being one sucker, it is made up of 6 suckers. So I believe these guys live up frog bladders. And no, I'm not going to go massacre some frogs just to see if I can find one of these. But yeah, that's where they live. This is one that's economically important because it's a pest of fish that are farmed. One species cost the farmed salmon industry in Norway an estimated 500 million euros, which is what, like $700 million or something like that. And these actually do enough damage that they can kill fish, not by themselves, but fish die because of secondary infections. They wound the fish enough that the fish get bacteria coming into the wounds. And you see that haptor at the lower left. That haptor is kind of triangle shaped, and it's got hooks around the outside and big hooks inside it for latching onto a salmon. The head is at the upper right. You can see it looks forked. That's just a pair of attachment organs that surround the mouth. And these can detach, so the worm can basically just kind of grub all the way around in a wide radius. These guys, incidentally, give birth to live young, and this one has a juvenile folded up inside itself. And the juvenile actually, you can't see it here, but there are cases where the juvenile will have another juvenile living inside it. So you can actually get 3 generations nested one inside the other like they were Russian dolls. Kind of a neat trick right there. And yeah, certainly economically important if you're in the aquaculture biz. And then I actually was able to track down a picture of Oculotrema hippopotami, which is a parasite of hippopotamus eye sockets. The paper on these was actually written by a South African wildlife biologist, or a pair of them, that actually had to go out and cull a hippo herd. Hippos are among the most dangerous animals in Africa. They look adorable, but they're notoriously bad-tempered, and they'll just come over and squash you like a bug if you get too close to them. They also have this fun thing where they will poop out a lot of liquid feces while helicoptering their tails and spread it in droplets all over you. Yeah, don't mess with hippos. And part of a hippo herd had to be culled. So they happened to shoot about 15 hippos. And, you know, what do you do with 15 dead hippos? Well, what they did was dissect out their eyes. And they found that 90% of the hippos were infected with Oculotrema hippopotami, which looks like this. The front end is red, the hind end is white, and the hind end has that haptor, which consists of a grouping of 6 suckers. So that's what these look like, and now you know. Even weirder is this thing. This is Diplozoon paradoxum. And what happens here is that larvae attach to fish gills. This species in particular, I think, tends to like European carp, if memory serves. And a larva will attach to a fish gill, try to survive, and hopefully it will find another larva. If it does, the two grow together, and they not only grow together, but their tissues fuse, their organs fuse, and they develop this kind of crossover reproductive system such that each one's sperm duct crosses over and lies right next to the other's egg duct, which means each one spends the rest of its life fertilizing the eggs of the other. What you're looking at is you've got a pair of, the mouths are up at the top left and the top right. They've got little accessory attachment organs next to them, not easy to see. Each one leads into a forked gut. And then in the bottom half, the haptors consist of a bunch of clamp-like structures, which is what you're seeing down there on those blunt ends. Those are little clamps. And the anatomy ends up working like this, where you've got ovaries up at the top and testes down at the bottom, and the ductwork between them crosses over so that each one fertilizes the other. And the weird thing is that if a larva does not encounter another larva, it will die and never reach sexual maturity. The only way for these to reach sexual maturity in the first place is to fuse together and do that crossover trick, which means that Diplozoon is the only animal that we are absolutely sure is monogamous. Most other animal species that are mostly monogamous will reproduce outside the pair bond if the opportunity presents itself. Ask Dr. McDonald sometime about extra pair copulations in birds. It's a pretty common tactic. Diplozoon is absolutely known to be 100% monogamous because they can't reproduce at all unless they have fused with a mate. So there, little food for thought, I suppose. Finally, Cestidaria are intestinal parasites of vertebrates. There's no gut whatsoever. You've got three major groups, not two. That's a typo. Amphilinids are not divided into segments, and they mostly live in the body cavities of fish and reptiles. Gyrocotilids, the body's not segmented, and these are parasites of chimeras. That's a group of fish somewhat related to sharks. They're sometimes called rabbitfish, ratfish, things like that. And then eucestodes are the true tapeworms in which the body's divided into segments. And I'll spend most of my time on the true tapeworms. We'll talk about those structures in a bit. Amphilinids look like this. This one's in the body cavity of an Australian long-necked turtle. There's not very many. I want to say there's 8 species of amphilinid known. Gyrocotilids typically have this, there's a sucker at one end, and then the sides of the body are ruffled. And gyrocotilids live in the intestines of chimeroid ratfish because somebody's got to. And this is what they look like, and they're not very well studied because ratfish live in the deep sea and nobody knows very much about them. So there you have roughly things living inside ratfish. Ooh, ah. And then here's a typical true tapeworm or eucestode. This is the pork tapeworm, Taenia solium. At the head, called a scolex, you've got various structures that attach the head to the lining of the gut of the host. In this species, there are 4 suckers. There's also a anterior projection kind of sticking out of the top of the scolex. It happens to be called a rostrum, and you have these hooks sticking out of the rostrum. So the hooks and the suckers attach the worm to the gut wall. Back of that, the body divides into a series of segments called proglottids. And these can extend, what is it? They found a tapeworm of whales that's something like over 100 feet of worm, almost all of it made up of one after another of these proglottids, of these segments. Now, this one uses suckers and it uses hooks. One that humans can get from eating raw fish is called Diphyllobothrium. This doesn't have hooks or suckers. It's got a pair of what are called bothria, which are kind of like groove-shaped suction cups. You can see one labeled B over there on the left, and this is what they look like. There was a recent concerted attempt to document tapeworm diversity that recently concluded with the discovery of 215 that were new to science. So on the right, you have Yorker izzardi, which, yes, was named for Eddie Izzard for reasons I'm not sure, which has 4 bothria, which have hooks on them. And then Letobothrium nicolai, I'm not even sure what that thing does. The one over there on the right. I assume that those are bothria right there, the kind of leafy parts. But yeah, that one just looks strange. So great diversity of attachment structure form in tapeworms, in particular ones in marine species. And the tegument is your basic one for Neodermata. You have those cytons, those cell bodies under the muscle layer, the ones that have the nuclei and most of the organelles. And they send up these projections through the muscle layers that spread out over the old ciliated epidermis and join together to form a continuous layer, a continuous syncytium. And unique to tapeworms, you have those microvilli called microtrichs on the outside of the tegument. They probably give the worm some traction, keep it from sliding out of the intestine where it lives. They also probably increase the surface area. Yeah, depending on what species you've got, they may take different shapes. This is a worm, I'm not even sure what it is, but it's got some of these microtrichs are called spinatrixes, and they're surrounded with phyllotrixes. And I think I first encountered spinatrix and phyllotrix as companions of Asterix and Obelix, which you may not be familiar with. Don't worry about it. Right. Anyway, the shape is important for classifying your tapeworm. Back of the scolex, back of that head, the rest of the worm is called the strobila. Same word as we encountered with reproduction of jellyfish. And the strobila may consist of a small number of proglottids, as it does in this one. Echinococcus is, I think that's normally a dog species, but humans have been known to get it. And proglottids kind of bud off the head end, and as new ones bud off, the old ones are pushed towards the rear. And as they're pushed towards the rear, they sexually mature. So at the back end of this echinococcus, you have a fully mature segment that's loaded with fertilized eggs, and that will then break off and the eggs will pass out in the feces. And then as you go from left to right, you go from the most mature segment to less mature segments, and then ultimately you get to the scolex, you get to the, quote, head end. In this species, you can see the suckers on the scolex. So this echinococcus consists of a small number of proglottids. On the other hand, Taenia solium, the pork tapeworm, and Taenia saginata, the beef tapeworm, may consist of quite a lot of them. This one is probably about 1 meter long and might consist of thousands of proglottids. But the basic idea is the same. Proglottids form at the back of the head, the back of the scolex, And as new ones form, old ones get pushed down towards the far end. And once they are sexually mature, they break off the far end and pass out in the feces. Some tapeworms, you'll have whole chains of proglottids will pass out of the feces at one time. I hope you've never had to deal with this too much, but we had a dog at the house once. He ended up going off to live on a farm. And actually, I'm not kidding. He really did go off to live on a farm. But when we got him, we fostered him for a little while. And he had tapeworms. And when he'd poop, which he liked to do in the house, you could see these, white wormy wriggling things that would come out with the feces and kind of thrash around for a bit. Maybe some of you have seen something similar. That would actually have been chains of multiple proglottids, which can actually be pretty active when they're fresh. In other tapeworms, the proglottids actually break up, and what passes out in the feces is separate eggs. The proglottids just kind of degenerate and disintegrate before they pass out of the host body. I think that's the case in Echinococcus, but it depends on what species you're working with. The eggs or the proglottids pass out of the definitive host, and then they have to get swallowed by an intermediate host. If this happens, the eggs hatch into larvae called oncospheres. Sphere, onco means a hook, right? You might know that the medical specialty of treating cancer is oncology. Onco is used for cancer because you think of cancer as something that gets its hooks in you. In fact, cancer is also Latin for crab, and people thought of tumors as being like these crabs that dug their claws into you and wouldn't let go. And oncology is the study of the thing with the claws or the hooks that latches onto you. And here, oncosphere means a sphere with hooks. And these larvae will pass through the body, and they may form a cyst called a cysticercus or a cysticercoid. Don't worry about the difference between them. When the definitive host eats the intermediate host, the cysticerci enter the gut, they hatch, and they develop into adults. So there is a sexually mature proglottid of the pork tapeworm, Taenius solium. Male organs in blue, lots of testes dumping sperm into a common vas deferens. Female organs in pink, where the ovaries drop eggs into a common oviduct with specialized yolk glands providing yolk cells to plaster around the mature eggs. Both of these passages share a common genital pore. Tapeworms can self-fertilize, but they will pair up and fertilize each other if there's two next to each other in the same gut. Once they've been fertilized, the other reproductive structures degenerate, and that uterus swells up enormously. And the shape of the uterus, once it's completely full of eggs, can be important for identifying species. Up at the top, Taenius solium is the pork tapeworm, and Saginata is the beef tapeworm. Both of these, by the way, infect humans. You get them by eating infected meat. And if you were trying to diagnose somebody with a tapeworm, you'd look for proglottids in a fecal sample, and this is how you might have to identify which tapeworm species the person had. So anyway, these will break off. Either the whole proglottid will pass out, or sometimes a whole chain of them will pass out. In other cases, the proglottid degenerates and separate eggs come out. Whatever happens, the eggs get consumed by the intermediate host. And they can look like this. That's the egg of a tapeworm called Hymenalepis diminuta. The definitive host of these are rats. Eggs like this pass out in the rat feces, and the feces get scavenged by beetles. And the eggs are actually surrounded by multiple layers of shell. The innermost circle is the actual larva. The rest is shell layers that surround it. And in the larva itself, you can actually see a few little things that look kind of like daggers. One is pointing more or less towards your 9 o'clock and one towards the 10 o'clock. If we could focus up and down on that, you'd see that that's actually 6 little spines that come in 3 pairs. And when this egg gets eaten by a beetle, the larva will break out, assisted by the beetle's digestive enzymes and jaws, and use these 6 spines to tear its way through the beetle's gut into the body cavity, where it will develop into a infective stage called a cysticercoid. In the case of pork tapeworm, the definitive host is people. People pass eggs out in their feces, and the feces get scavenged by pigs. Pigs don't deliberately go out of their way to eat human poop, but pigs are not always very careful about what they eat. And the eggs will enter the pig's body, and the eggs transform into, or the eggs hatch into larvae that travel through the pig body and form infective cysticerci in the muscle. Up at the top, that's a sample of pork that's got cysticerci embedded in it. Meat inspectors call this measles. So you have pork measles up there. They'd also have to look for beef measles in the case of the cow tapeworm, Taenia saginata. This is the kind of thing you would have to look for and be able to find if you are a USDA meat inspector. Incidentally, if you're worried about catching these, I mean, meat in the US is mostly pretty well inspected, but this is one reason why people don't eat rare pork, is that there's a risk of getting pork tapeworms from this. Cook your pork and you won't have any problems. Anyway, down below is a cysticercus that's been removed from the meat and stained. And what you're looking at is a little scolex that's kind of tucked inside the sac-like cysticercus body. And when a human swallows a cysticercus, the scolex pops right out, latches on to the definitive host's intestinal wall, that's you, and begins growing a new string of proglottids, and will grow in you. And if you get 2 of them inside you, they might mate with each other, and fertilized eggs will pass out of the body, and round goes the great circle of life. There are some where the hexacanth develops into a great big cyst called a hydatid, and each hydatid contains many cysts that bud from an inner layer, and every cyst develops into many scolices. So if you're unfortunate enough to eat a hydatid, the hydatid contains lots and lots of cysts. In fact, these cysts ultimately rupture and produce lots of little scolices that make this deposit in the hydatid called hydatid sand. And each one of those scolices, if swallowed, can pop right out and convert into a tapeworm scolex that will promptly grow new proglottids and become a new mature tapeworm. Right. Once the tapeworm's in the definitive host, it grows proglottids, and those pass out of the host anus. This is a drawing from 1850 by a great Japanese artist depicting what happens if you eat raw salmon. There are tapeworms in fish that can infect humans. This man has got a case of Diphyllobothrium nihonkaense. This particular one, the proglottids stick together and will pass out in long chains. In others, as I've mentioned, the proglottids pass out singly, or they don't pass, they disintegrate, and it's just the plain eggs that pass out. If this is a concern, by the way, fish that is sold in the US for sushi, by law, has to be flash frozen. This is not the kind of thing you could do in your home kitchen. The fish is basically blasted with liquid nitrogen, and fishing boats have special equipment for doing this. And flash freezing for, I believe it's 72 hours, is enough to put an end to the parasites. So if you're eating at a sushi restaurant, the fish has by law been flash frozen and thawed. That's not supposed to affect the taste or the texture, but it is supposed to be enough to kill the parasites. Now, if you're in Japan, they still like eating very fresh sushi. My wife's father was a commercial fisherman, and he used to get on the boat with his wasabi and soy sauce. I mean, he would eat sushi about as fresh as it was possible to eat it. Like, as in, we just pulled this over the side. And I don't know if he ever got parasites, but fresh sushi like that is a potential risk. But the sushi that you get in our local sushi restaurants has all been flash frozen, and you can't catch anything from it. So there, I will now make this go away. Tapeworm infection, by the way, causes weight loss. Tapeworms don't have guts of their own. Just sitting in your gut, they absorb digested nutrients directly through the skin. And since they absorb them, you don't absorb them, which means that if you've got a tapeworm, you'll start losing weight. Mind you, you can also end up malnourished with B vitamin deficiencies and anemia. But tapeworms were actually once sold as a weight loss aid. If you eat tapeworm cysticerci, and they'll pass into your gut, they will hatch out, and they will absorb all of those, they'll absorb all of that fat and sugar that you're taking in. They will absorb the enemy that is shortening your life, and your body won't. So you can eat whatever you want to, and you'll still stay slim. Of course, you'll get sick, but the important thing is you'll be thin. So yes, you might be interested to note that people were just as stupid about weight loss 100 years ago as they are now. Occasionally this gets mentioned today. Tyra Banks actually promoted the tapeworm diet on her talk show back in 2009. You can actually find people who claim that deliberate parasite infection is actually good for you because it gives your immune system something to do, which means your immune system is less likely to turn on you. If your immune system starts attacking parts of your healthy body, you've got an autoimmune disease. There's a theory out that suggests that the reason we get autoimmune diseases is that before civilization, everybody had parasites and we'd adapted to cope with it, and we had immune systems that could keep them in check. Now that sanitation and things like that mean that most Westerners don't have parasites, the idea is that the immune system no longer has anything to do, so it kind of turns on its own body. And I am not able to evaluate this. I'm not a medical doctor. There are apparently some encouraging clinical trials of deliberately infecting people with parasites. I'm certainly not going to recommend it, but it is something that's out there that you might encounter. Almost done. If the definitive host eats tapeworm eggs by mistake, remember that you have to go from an intermediate to a definitive host. And the definitive host gets infected by eating Cysticercus larvae. If the definitive host eats not Cysticercus larvae but eggs, the eggs will hatch and transform into Cysticercus larvae, and they can end up in places you really don't want them, like the brain. There you've got cysticerci that have formed inside a human brain. Each one of those little round things with a P inside is a cysticercus. You can also get cysticerci in places like muscle tissue. I don't know if they cause severe problems unless there's a whole lot of them, but what they'll do is calcify. So you'll go in for an X-ray and there'll be a bunch of little bright dots buried in the muscles surrounding your bone, visible on an X-ray. Echinococcus is particularly dangerous because if you eat an egg, it'll form a hydatid, one of those giant cysts that buds off, little cysts that buds off lots of little scolices. And this is what one of these looks like on a CAT scan. I've actually seen footage of neurosurgery to remove one of these things, and removing them is very tricky because they're very soft. They will easily rupture, and if they rupture, the fluid is extremely allergenic. The fluid inside one of these things causes a life-threatening allergic reaction. So you've somehow got to get out a very delicate little water balloon type thing without having it rupture. And this is not very easy, but you can actually find some footage of the operation on YouTube, or you could last I looked. So yeah, pretty good reason to be careful about not ingesting tapeworm eggs by mistake, because you could end up with that. You need that like you need a hole in your head. That was a joke. Okie doke. Go back, see what's on the chat. I owe you Lab 6, and life is going to be really annoyingly weird this weekend, but I will try to finish Lab 6 and get it to you as soon as I can. And that's another one you should be able to finish without too much input from me. I'll go ahead and stop recording, and then I'll answer this question.